Good day. My name is Paul Wax. I'm the Executive Director of the American College of Medical Toxicology, and I'd like to welcome you to our webinar series entitled Medical and Public Health Considerations of COVID-19. Today, we will be featuring a webinar on vaccine safety. Next slide. I'd like to thank our webinar series partners for helping to promote our webinar series. Much appreciated. Next slide. This webinar is in a series of most Wednesday webinars. We've been doing this now for about 5 months, or 6 months. And all the webinars are recorded and posted to the ACMT webinar page at www.acmt.net/COVID-19 web. And this webinar will be recorded as well and will be posted on Friday, and we'll let you know by email as soon as it's posted. We also have constructed a certain number of FAQs for the webinars, and those are also up on our website. We will also have a Q&A at the end of this presentation. Next slide. For the Q&A, please chat in your question either into the Q&A box or into the chat function. For those of you who have come in on the YouTube, you can use the chat function, and we'll try to get to as many of your questions as possible. Next slide. I'd like to thank my co-moderator, Dr. Ziad Kazi. Dr. Kazi is a member of the Board of Directors of the American College of Medical Toxicology and president of the Middle East and North Africa Clinical Toxicology Association and an associate professor of emergency medicine at Emory University. And next slide. I'm delighted today to introduce our guest moderator, Dr. Keith Burkhardt. Is a past president of the American College of Medical Toxicology. Currently, he's senior advisor for medical toxicology in the Center for Drug Evaluation and Research, Division of Applied Regulatory Science, of the Office of Clinical Pharmacology at the U.S. FDA. And Dr. Burkhardt will introduce our plenary speaker. Thank you, Dr. Burkhardt.
Paul, I really appreciate the opportunity to moderate this session. So timely and really the topics on the mind of the entire country and the world right now with what's happening in the research that's going on right now. So it's my pleasure to introduce Dr. Narayan Nair. He works at the FDA as the director of the Division of Epidemiology in the Office of Biostatistics and Epidemiology in the Center for Biologics Evaluation and Research. He received his undergraduate degree at St. Louis University and his medical degree from the Uniformed Services University of the Health Sciences in Bethesda, Maryland. Subsequently, he became board certified in internal medicine. And in 1990, he was commissioned as an officer in the U.S. Public Health Service until his retirement after 30 years just last year. He had a number of interesting assignments through this time. He worked in his clinical practice was at a community health center in Pennsylvania and also with the Indian Health Service in Arizona. One of his great assignments, and that very near and dear to those of us in medical toxicology and emergency medicine, he was assigned to the Office of the U.S. Surgeon General for disaster preparedness. And actually, I met and worked a little bit with Dr. Nair when he was at FDA's Office of Counterterrorism and Emergency Coordination, working as part of a team for countermeasures for biologic, radiation, and chemical threat agents. And I do have to say one of his other assignments, as our thoughts and prayers go out to those being impacted by Sally, he was part of the medical response team for Hurricanes Katrina and Isaac, and had other assignments with the medical response for inaugurations of some of our past presidents. And with that, I'd like to turn it over to Dr. Nair and now to teach us and talk to us about the vaccine adverse event reporting system and how the FDA monitors vaccine safety. Take it away, Dr. Nair. Thank you.
So thank you so much, Dr. Burkhardt, for the introduction. And thank you for the opportunity to speak to you. And I know everyone's extremely busy, so I appreciate people taking the time to learn about this topic. Here is my disclaimer. The views and thoughts and opinions that I'll be discussing are solely my own. They don't necessarily reflect the official views or policies of the government, the Department of Health and Human Services, or any of the operational divisions. And, just to give an overview of my talk, I was briefly going to go over the organization of the FDA. I was going to discuss the phases of drug and vaccine development. And then I was going to talk about vaccine safety and post-market surveillance options, including VAERS. So the FDA is organized into centers. There's a center that evaluates drugs, a center that evaluates tobacco, center for food, center for medical devices. I work in the Center for Biologics, Center for Biologics Evaluation and Research, or CBER, and our head is Dr. Marks. And we work on vaccine and biologic products and blood products and assessing their safety and efficacy. And you can see it's comprised of a number of different offices. I'm housed in the Office of Biostatistics and Epidemiology. And our role involves evaluating the safety primarily for my division in the post-marketing stage, and I'll go over that a little bit more. And so this slide illustrates the safety in the life cycle of FDA-regulated products. So this is for drugs or vaccines. And essentially, it starts in the preclinical phase. Before it's tested in humans, there's a lot of assays and laboratory work and work in animals where the safety and biological activity is assessed. Then it moves on to phase 1. This is the first time it's used in humans, and it's under very close monitoring and controlled conditions. All these clinical trials are done with informed consent under ethical considerations. And for phase 1, really the goal is to see whether this product is safe in humans and what is a safe dose. Typically, there are smaller numbers of people in a highly monitored setting. And if it passes that phase, it goes on to phase 2. Phase 2, again, the safety is monitored very closely, and this is the phase where they also evaluate efficacy, whether this product works. So in the case of a vaccine, is it generating an immune response? And these are somewhat larger numbers than phase 1. Phase 3 are the large-scale trials. These are sort of the pivotal trials that are used by the FDA. We look at all the trials, but the phase 3 ones are the pivotal ones. We look to see whether this product should be approved or not. And again, the safety is closely monitored and the efficacy as well. And you can see that all these phases and feed into the safety below. After phase 3, if the product is successful, it is approved and it is used across the country. But the monitoring of safety isn't complete. We do post-marketing safety surveillance, and we evaluate how the product is performing from a safety standpoint after it has been approved. And so using all these sources of information, we evaluate the safety, and we come up with strategies and actions to minimize risk. The whole point of the vaccine and drug approval process is to have a product that has a favorable risk-benefit profile. No product is 100% effective. No product is 100% safe. But what we want is a product that maximizes the benefit and minimizes the risk. And so that's what we're striving for. And depending on the condition, that risk-benefit profile may look very different for someone who has end-stage cancer. It may be reasonable to tolerate a level of risk for a treatment, as opposed to a vaccine where we're giving it to healthy people. And so the risk-benefit profile really depends to some extent what we're treating. So I'm not going to delve into a huge amount of detail, but I did want to cover the COVID-19 vaccines in phase 3 trials in the United States. And the source of this is clinicaltrials.gov. If you want further information, there's dozens of trials related to COVID-19. They're posted on clinicaltrials.gov, and a lot of information about what the trial design is, if you're interested in volunteering for a trial, if there's a site near you. That information is all at this website, and it's easily accessible and searchable, not just for COVID, but for many other conditions as well. And these are the 4 vaccine candidates that are being studied in the U.S. There's many others that are going to be studied and that are currently undergoing evaluation in other countries. These are just the ones in the U.S. So by no means am I endorsing any of these or saying these are going to be the first out the gate. I don't know, but these are the ones I wanted to highlight. And, you know, as you recall from your basic biology, the goal of the vaccine is to stimulate an immune response to an antigen on the virus or bacteria. And so there's different platforms that can be utilized to accomplish this. The first one on this list, the Moderna mRNA vaccine, uses a mRNA particle to elicit this vaccine response. As you recall, the SARS-CoV-2 vaccine has this spike protein on the surface, and that is what triggers the antibody response. And so for the goal for these vaccines is to promote that antibodies to this spike protein. And the mRNA vaccine, which include the Moderna and also the one after it, the Pfizer BioNTech candidate, they use an mRNA particle. That mRNA particle codes for that protein, spike protein, and then that would generate a immune response. The next two vaccines on here, AstraZeneca and Janssen, these candidates are also in phase 3 trials. They use a little bit different approach. They use a viral vector to basically, this is a virus that is harmless to people, that has been genetically modified so that it exhibits that spike protein on its surface. And so when the vaccine is injected, it triggers not only antibody response, but a cellular response as well to build immunity. So that is a very kind of thumbnail basic summary of the vaccine candidates that are in phase 3 trials. Many of you may have heard the AstraZeneca vaccine was being studied in the U.S. and several other countries in phase 3. And these clinical trials, the safety monitoring is very tight and very thorough. They did have an individual that suffered an adverse event. It was reported to be transverse myelitis, which is a autoimmune condition, inflammation of the spinal cord. There's no way to know if that's at this point if it was related to the vaccine, but in you know, as they do with these studies, they brought it to a halt to do further evaluation to see whether there's any possibility if it's related to the vaccine. So I believe those trials have resumed in other countries. I don't believe it's resumed in the U.S. at this point. So I've sort of highlighted the pre-licensure, where before it's given in humans, safety is evaluated in a laboratory setting. In the clinical trials, these studies, typically the COVID-19 vaccine trials, involve as many as 30,000 people for each candidate vaccine, 15,000 in the placebo arm, 15,000 receiving the vaccine. It's randomized, blinded, and they're evaluating the safety and efficacy. So with all that data, why do we conduct post-market safety surveillance? And really, it's because the clinical trials, although it provides extensive data, that there are limitations. And a clinical trial may exclude groups within the general population who later are exposed to the product. Also, with a clinical trial, you're doing a post-hoc safety analysis. Typically, you like to identify your statistical analyses before, but with a clinical trial, if a safety signal arises, sometimes you must do a post-hoc safety analysis, and that's fraught with some issues in terms of you may get something spurious. If there's a side effect, an adverse event that occurs maybe a couple of years later or even 7 to 8 months later after receiving the product, if it has a long latency effect, it may not be detected in these clinical trials because clinical trials sometimes don't run that long. So those are all limitations. And also, post-marketing safety surveillance is a helpful way. Once these vaccines are studied or drugs are studied, they have to be manufactured. And if there's a production problem, if a certain lot has a contaminant, we can detect that with a focused warning system using post-market safety surveillance. So this slide sort of illustrates another limitation of clinical trials, and you may need large sizes to detect the adverse event rate. So in this study, you can see that it's comparing the rates in the background population, and just hypothetically, let's say we're talking about headaches. Let's say 5% of people just get a headache in a particular time frame versus 10% of people who receive a drug or a vaccine. So it does increase the risk. It doubles the risk of getting that headache. And the sample size to detect something like that would be 950, which is certainly doable. And then if it were to triple, you could see that the sample size would be 320. Where you have issues, if you get into a very rare event, something that occurs rarely in the general population, and although the risk is increased with the product, you still need a very large sample size. So from 0.2%, if that's the background rate, to increase it to 0.4%, then the sample size you would need is 25,000. And as you can see through this table, when you talk about really rare events, 0.01%, to detect an increase to 0.03%, you need 175,000, or if you double that from 0.01% to 0.02, you have 500,000. And you can see sort of the statistical caveats. This is assuming a two-arm trial. But clinical trials, typically aren't this size. So they may not detect these very rare events. So what do? We have the clinical trial data. So we have the sponsors submit a pharmacovigilance plan, and we review that very closely. So when they are applying to license their product, to sell their product in the United States and make it available, they submit this pharmacovigilance plan. What are their plans to monitor safety after it's released? And they discuss if what's available, what do we know about the product? If it's approved elsewhere, what do we know about it? Are there similar products that we can maybe extrapolate safety issues? During the clinical trials, were there safety issues identified? So we look at all this and develop a plan to monitor safety. And the pharmacovigilance plan allows us, informs us on post-marketing studies. It allows us to really consider whether we want to do any kind of post-marketing regulatory action, and allows the discussion of whether we want to do active surveillance post-marketing studies. And I'll talk a little bit about that later on in my talk. So pharmacovigilance is really looking for signals. And this organization, the Council for International Organizations of Medical Sciences, defines a signal as information from one or multiple sources, which could suggest a potential causal association or something new about a known association. And it's of sufficient likelihood that it may need to be verified. And so signals can come from pre-marketing data before a product's approved, or even can become from preclinical data, or it can come from events from other products in the same class. And so this slide sort of illustrates the approach for post-marketing safety and safety in general for these drugs and vaccines. And basically the way we think of it is we have successive layers of defenses. Now each layer has its limitations, but the goal is with each layer being somewhat different in terms of limitations, that we'll be able to capture safety concerns by building one layer upon the other. So certainly the first layer would be the clinical trials before the product is approved, and then other layers would be passive surveillance, receiving adverse event reports, and then active surveillance. And then lastly, post-marketing studies. So using all these tools, our goal is, again, we're looking to maximize the benefit and minimize the risk of these products. So this slide goes into a little bit of more detail of what I just described and talks about the tools. And so our sources of data for safety are these clinical trials, pre-licensure, and then once a product is licensed, spontaneous reporting, and I'll talk more about that, and that includes VAERS, or large electronic healthcare databases, and then other sources such as literature or foreign regulatory agencies that provide us information. And all of those sources, we can generate a hypothesis. If we see a signal, if we see some adverse event that doesn't seem like it's normal, it seems unusual, that it might be related, it's just a hypothesis, but we can test that hypothesis using various verification tools, including additional clinical trials, observational studies, or these large electronic healthcare databases. Ultimately, we've generated a hypothesis, we've tested the hypothesis. Based on that, we may take a regulatory action, which could include communications, labeling changes, and recalls. So when we talk about safety surveillance, we talk about we talk about passive surveillance. And basically, what passive surveillance includes are spontaneous reports from providers, patients, and consumers. And these are submitted directly by the sponsor or directly to the FDA and CDC. And the tools that we use are the Vaccine Adverse Event Reporting System, VAERS, or the FDA Adverse Event Reporting System, PHARES. Both these tools, PHARES is used for drug products, and biologic products, vaccine products are submitted. They use VAERS to report adverse events. So this is passive surveillance, where we receive the data from outside. And just to provide a little bit more detail about VAERS, it's co-administered by the FDA and CDC. The forms are submitted by companies that gather data, healthcare providers, parents, patients, anyone can submit data. And there's a contractor that enters the data and codes it. We use a MEDRA coding system so that allows us to search for certain adverse events and search for trends. We receive 50,000 reports annually. 20% are serious. Now, bear in mind, these are spontaneous reports, so it doesn't assess causation. It's just a hypothesis generating. So when we talk about these serious reports, we're talking about patients who are hospitalized, have a prolongation of a hospitalization, congenital defects is also considered serious, or have some type of life-threatening situation. So serious adverse event reports, we review those manually. Our medical officers take a look at all of those to assess whether there's any type of signal or assess whether any action is needed. Non-serious reports, we typically assess those through data mining. We do a statistical analysis to see whether we're seeing an increased proportion of any adverse event on this product related to other products. And this slide shows the VAERS forms. The form can be submitted electronically, can be submitted through a fillable PDF, it can be submitted by paper. And the sections that are highlighted are sort of the essential sections that are needed. People don't need to fill out the entire thing, although it's very helpful if they do. But the highlighted ones are really the ones that are needed to generate a report. So passive surveillance, very useful. As I mentioned, can generate a hypothesis. It's open-ended. We aren't looking for anything in particular, so it's not a narrow type of data. We receive all types of data, and we can detect new or rare adverse events. Geographically, it's very diverse. Anyone in the U.S. can submit a report, and it helps us, as I mentioned, monitor production lots. If we have that lot information, we can do a search and see if we have any concern about any type of production issues. But passive surveillance has limitations. We can't calculate incident rates with passive surveillance for two reasons. One is underreporting. All of us are busy. We may not have time to submit a report if our patient has an adverse event. And secondly, an individual may decide not to report themselves. So we have underreporting. We have an absence of denominators. We don't know how many people actually are receiving this vaccine or drug. We can look at distribution data, but ultimately we don't have firm numbers of how many people are receiving that product. So we don't have the denominator, and we really don't have the numerator because of underreporting. The opposite of underreporting can also arise where we have reporting bias. We call this stimulated reporting oftentimes can occur if there's publicity. If someone goes on the news and says they had this side effect from this vaccine, or there's some type of litigation, if it's a newer product, oftentimes we'll see a surge in reports after those type of things, and it may not necessarily mean that those reports are occurring more frequently than they were previously, but it just could be an indication of what we call stimulated reporting. Someone was prompted to report based on what they saw or heard. We don't have a control group or untreated, unvaccinated group. So, with those clinical trials, we have a placebo group, the vaccine group. Once the data is unblinded, if we see 5 people had a severe rash that received the vaccine, that might be concerning. But if our placebo group, we see 10 people had a severe rash, then that might be reassuring. So you can do these comparisons in clinical trials. We can't do that in passive surveillance. These reports may have missing inaccurate data. We're just basing on what someone is submitting to us. Oftentimes we're not able to get the diagnosis verified. So we can't assess causation. Again, as I sort of emphasized before, they generate a hypothesis. So once we have the hypothesis, we can move on and do something a little bit more firm to verify active surveillance. And we can use healthcare data collected by large insurers and healthcare organizations. Usually these are collected for billing or electronic medical records. And we can leverage these existing data by partnering with data holders, insurance companies, databases, the owners of electronic health records. And so we can use this and identify outcomes and do studies. And the advantages are this is standardized. It can be accurate because it's typically coded. There's consistent data collection. We do have denominators and numerators because we have the records. The disadvantage is, as you would imagine, these studies cost a significant amount. There's maybe time. It takes a long time to do these. They're limited in scope. We have to identify what outcomes we're looking for. So if there's something unexpected, we may miss that. Also, automated codes may be inaccurate as well. Although certainly there's a higher degree of accuracy you would think than spontaneous reports. Still, automated reports may be inaccurate. And so these are the CBER post-marketing surveillance systems. I won't go into a huge amount of detail, but they can involve tens of thousands, in some cases millions of healthcare records that are aggregated and analyzed. Sentinel program is a partnership with Harvard Pilgrim and looks at claims. The CBER BEST program, it looks at claim data and some electronic health records data. And then there's partnerships we have with the Center for Medicare and Medicaid, the VA, and a program that's coordinated by CDC, the Vaccine Safety Data Link. And all of these are essentially reviewing data from electronic health records or claims data. So we've talked about sort of identifying a signal, verifying a signal. Once we have a signal that we verified and we think that something is going on, what are our options? Based on the signal, we have a number of different things that FDA can do. We can change the package insert. We can add something to a label. We didn't know this side effect existed when it was approved. We're going to change the label and make sure that providers are aware of it. On the FDA website, there's something we call a Section 921 posting. When we initially are looking at a safety issue, we post that on the website to make the public aware. And then when we've sort of resolved that, we also post that on the website. And so that's a good source of information for a particular product, what we're worried about. If we want to post a little bit more detail, we do a public health notification and release the data that we have about the safety issue. We can present at meetings such as this and submit abstracts, again, so that we can make that data available. And then, if we have a high degree of concern, we can do a Dear Doctor letter or Dear Healthcare Provider letter where letters are mailed out to providers to make them aware of a certain issue with a product. We have, and this isn't typically used for vaccines, but we have something called a risk evaluation mitigation strategy. For certain products, and in CBER, we've used this for oncology products that have a significant amount of toxicity. We put in a program in place where healthcare providers certify that they're aware of the toxicity and how to manage people with the toxicity, and certain sites have to certify. Not as relevant, we haven't used that for vaccines. If we're concerned about a manufacturing issue, we have FDA inspectors who can go out to the facility and conduct an inspection and make sure that good manufacturing practices are being enforced. If we're very concerned, certain times we get data and it just shows us that the risk-benefit profile is shifted. The benefit does not outweigh the risk. And at those times, a product may have to be withdrawn. And then also, we can conduct post-market studies. Either we can ask the company to do post-market studies. If we find a new safety issue, we may ask the company to evaluate it and do a formal study, or we can use our active surveillance tools and do a study ourselves. So in conclusion, vaccine safety, I want to emphasize, it's monitored throughout the product life cycle, from when it's first in the laboratory in the test tube to when it's first injected into a person to when it's sold to millions of people, even if it's a vaccine that's been on the market for decades, that we are continually monitoring the safety. And this will be the case with any potential COVID-19 vaccines. we use active and passive surveillance tools to monitor the safety of vaccines after approval. And then lastly, if you're interested, this process of approval of vaccines is quite transparent in most cases. And so I would sort of highlight a couple of resources for people that want to learn more. One is the CDC has an advisory committee on immunization practices. They're the committee that meets 3 times a year and says, we recommend all school children get this vaccine at this period of time, at this age. And so they issue recommendations periodically. I'm sure most people have seen those. They've held a series of meetings about COVID-19 vaccine candidates and what to look for, including vaccine safety. And these are subject matter experts. They're not part of the federal government. It's an outside committee that advises the federal government. And so I think that's a very good resource for people that want to learn more. Their meetings are live streamed and the public can participate in the streaming and ask questions. They have a public comment section. The FDA has an advisory commission that advises on vaccines as well, the Vaccine, Biologic and Related Products Advisory Committee. And they are scheduled to meet in October as well, and they'll be discussing COVID-19 vaccine candidates. Most new vaccines are discussed in that forum, and the data is shared in a public venue. So those are a couple of resources for people to visit if they're interested. And as I mentioned, those meetings are live streamed. The meeting materials typically are posted 2 to 3 days before the meeting occurs, and past meetings as well are posted on the internet. And you know, in conclusion, I wanted to thank everyone for their attention and for joining me. I believe the questions are at the end of the meeting. I'll be happy to stick around to answer those. And I particularly wanted to thank all the people who took time out of their busy schedules to join, particularly healthcare workers, if you're a frontline worker or involved in public health at all. I'm so grateful. We all are so grateful for everything that you do. And that's all my remarks. Thank you.
Thank you so much, Dr. Nair. That was a great talk, really going into and detailing all the sources of data that the FDA receives, seeks out to ensure vaccine safety and the actions that sometimes the agency takes in that. Great talk. Thank you so much. As Dr. Nair mentioned, the Q&A is going to be at the end after our next speaker. So please keep putting your questions in either the Q&A box that you have or the chat box, either of those, and we'll do those, answer those after the next talk. And with that, I am going to turn it over to Dr. Ziad Kazi to introduce our next speaker. Thank you.
Thank you, Dr. Burkhardt, and thank you, Dr. Nair, for a wonderful presentation. As we are anxious to learn more about the vaccines for COVID-19, and as millions of people are anxious to get vaccinated to hopefully resolve this pandemic challenge, it's important to look back at history and look back at our past safety of vaccines. And it's our honor and pleasure to present to you Dr. Daniel Shepshelovich, who is one of the senior authors on a recently published study in Annals of Internal Medicine titled Postmarketing Safety of Vaccines Approved by the U.S. Food and Drug Administration, a cohort study. This study was published in September 2020, and Dr. Shepshelovich is a senior author and researcher who's coming to us from the Sourasky Medical Center in Tel Aviv, Israel. We appreciate you doing this live at 10:30 p.m. Local time in Tel Aviv. Dr. Shepshelovich, the floor is yours.
Thank you. Thanks for having me. Yes, we did a study focusing on the of vaccines approved by the FDA, and it was published recently, very timely for us. Yeah, so my talk basically is about data supporting the presentation which we've just heard from Dr. Nair. So we think that public trust in vaccine safety is very important because we need the public to agree to vaccinate themselves and obviously their children. And we know that rates of vaccinations have been decreasing a bit in past years, maybe differently in different countries. And many of us have seen outbreaks. I personally have seen outbreaks of measles and other diseases I was not sure I'll ever see again because of unvaccination. So it's very important for us as a medical community to uphold the safety of vaccines as they're perceived by the public. So we've just heard how hard it is and how tough it is to get a new vaccine approved, and how important it is still to have post-marketing surveillance. As we've just heard, for rare adverse events or long-term complications, or for adverse events in populations not represented in the approval studies. And what we aim to do is basically to describe and analyze all of the post-marketing safety-related vaccine adverse events. And a secondary aim was to see where was the data coming from, if there was any new safety data, which of the post-marketing safety surveillance programs we've just heard described, which of these actually provided any new data. So what we did, we used data provided by the FDA. The cohort used in our study was all the vaccines approved through a 20-year period. We wanted to use all of the vaccines, so we will have an unselected cohort. We've identified all of the vaccines and the FDA, and we've identified all of the vaccine labels, the first vaccines, the first labels approved by the FDA, and the last labels until January 1st, 2020. We collected data for vaccine characteristics, the indication, vaccine type, when was it approved, and what kind of data was used to support the initial approval. So sample size, study design, was it placebo controlled, or was it controlled versus an existing vaccine, and how long was the safety follow-up period. And for each vaccine, basically, we compared the most recent label to the initial label for any safety-related changes. And any change, we looked at what was the regulatory type, whether it was a black box warning, a contraindication, a warning and precaution, when was the change made, specifically how long after the initial approval was the safety issue identified, what was the safety issue, and what was the source of the safety issue. So basically, this is a summary of our results. There were 57 vaccines approved during those 20 years. 25 of them had any kind of safety-related label modifications, and 32 had no safety-related modifications at all. Basically, we see that there were almost no changes between the two groups, maybe except influenza vaccines, who are, tend to have less safety-related label modifications, perhaps because it's a very similar vaccine again and again. Other than that, no other major difference between both groups. It is interesting to see that the vast majority were approved based on randomized controlled trials, pretty large trials, median sample size of more than 4,000 people. Most were compared with other vaccines and not placebo. Median safety follow-up were approximately a month and a half, so maybe a bit short on this point. In this cohort, there were 58 post-marketing safety-related label changes, 49 warnings and precautions, 8 contraindication, and 1 safety-related withdrawal I will address in the next slide. Median time from approval to safety modification was 5 years. Now, that may sound as long. We did a similar study in drugs and a similar study in medical devices. So for drugs, for medications, it was 10 years, and for medical devices, even a bit longer than that. So relatively a pretty good results here also for vaccines. Most safety issues were not very significant from a clinical point of view. So the most common was expansion of population restrictions, meaning specific populations which perhaps should not get the vaccine. So immunocompromised patients, people with specific medical conditions, premature infants and pregnant women. 13 safety modifications related to allergies, almost all of them to latex containing packaging. So not very major issue here as well. 12 modifications were post-vaccination syncope, 4 modification infections transmission, mostly rotavirus, 3 neurologic complications, febrile seizures, and 1 Guillain-Barré syndrome, and 2 warnings and 1 withdrawal related to risk of intussusception in rotavirus vaccines. So 6 vaccines were withdrawn for commercial reasons, or sales or obsolete products. The best example perhaps was the pneumococcal vaccine, 7-valent conjugate vaccine withdrawn when a newer and better vaccine was introduced. And neither updated or Google search didn't find any safety reasons for these vaccines to be withdrawn. The most common source of the safety data was Postmarketing Surveillance, VAERS program, just described by the previous speaker. And the single vaccine withdrawn was RotaShield for rotavirus infection back in 1998. It was withheld less than a year after marketing. The FDA actually became aware of potential safety issues less than 2 months after the vaccine was approved. Again, this relates to a slide presented by the previous speaker. The extra prevalence of intussusception for people with this vaccine was between 1 in 5,000 and 1 in 10,000 infants. So it's kind of obvious why this was not described in the studies supporting this vaccine, which the sample size was approximately 8,000 people, and obviously not powered to detect such small differences. However, the VAERS system detected it almost immediately, approximately 2 or 3 months after approval, underlining basically how effective it is. Other data was scientific publications, mostly investigator-initiated, reassessment of prior data, and again, the latest allergy. So a lot of vaccines, we had no clear indication of the safety data, but a lot of them seems to be expert opinion or just common sense, like not to vaccine with live vaccine immunocompromised patient pregnant women, or not to give rotavirus vaccine for infants with past episodes of intussusception. So basically, in conclusion, a large cohort of vaccines over 20 years found to be incredibly safe, much safer than medications, much safer than medical devices, much safer than basically any other medical intervention, again, because it's meant to treat healthy infants. And the only safety issues which was really clinically significant and caused withdrawal was detected by the existing post-marketing surveillance system almost immediately. So I think we can say that we have data to support that both the FDA approval process for a vaccine and the current FDA surveillance program are excellent. The only thing we could, when analyzing the data, we can think maybe can be improved is that the current system does not provide enough data for special populations for pregnant women and preterm infants, immunocompromised people. And this can be achieved either by specific pre-approval studies for these populations or by specific post-marketing surveillance of these populations, just to make sure that the new vaccines are safe for these people as well. So as I said, when we started the study, we were looking for, as we always do when we do this kind of studies, for points of weakness, for things we maybe can contribute and say, hey, maybe we can do this better. But for vaccines, basically, the system works great, both the pre-approval system and the post-approval system. Thank you so much for listening, for contributing with your time, and thanks again for inviting me.
Dr. Shepshelovich, please stick around for questions, and I would like at this point to turn it over to Dr. Burkhardt as well as Dr. Wax to moderate the initial part of the discussion.
Thanks, Dr. Kazi, and it was a wonderful talk and really great follow-on, and highlighted and brought up some points that are related to a number of the questions that we're getting in that here now. And one of those, which you touched on, was what is the typical duration of a phase 3 vaccine trial? I seem to catch that was about a month and a half. So there's a number of questions asking, stating that the coronavirus trials that are going on right now are really happening at a rapid pace and seem like they're coming on board a lot quicker and that. So, Dr. Nair, would you be able to comment a little bit further on that and how this seems to be happening so fast compared to some of that previous history?
Yeah, sure. Thank you for the question. And can you hear me okay? Am I coming through all right?
Yes, you're fine. Thank you.
Okay. Yeah, so I'm sure people have heard of Operation Warp Speed, And if you go to one of the slides in my presentation where I sort of went through those phases, Operation Warp Speed doesn't necessarily skip over any of those phases. So there still is the preclinical work, phase 1, phase 2, phase 3. How they compressed it and made it faster is that, most vaccines are developed by private industry, in some cases a partnership, but oftentimes, these phase 1 studies, to move on to phase 2, a significant amount of funding is needed. And so there's a lag between phase 1 and phase 2 that involves really raising funds. And between phase 2 and phase 3 studies are very expensive. And so there's, again, a time frame, an interval where companies are maybe trying to get one of the larger companies to sponsor these phase 3 trials. And lastly, the manufacturing process, to step that up and ramp that up, all of that occurs after the product is approved. No company wants to invest in that if they have a product that ends up not getting approved. So what Operation Warp Speed did is my understanding, and I'm not an expert in it by any means, but it compressed the time frame by still doing those studies, but not waiting. They took out that funding component by having the government assume the risk and the government stepping up manufacturing, even if it ends up the product's not approved, to sort of get rid of those delays in between. But the studies themselves are still being conducted, and they're looking at endpoints that have been used for other similar vaccines. So in terms of how long a phase 3 study goes, in general terms, it depends what the endpoint is. For efficacy, they may look at 6 months and compare the placebo group to the vaccine group, and in terms of how many people got sick in the placebo group and how many got sick in the vaccine group. But for safety outcomes, they may follow those people for up to 2 years. Now, that doesn't mean the FDA will wait for 2 years to approve it, but that means that after 2 years, they'll gather that data, and if needed, add that to the label if the product has been approved and so forth. So I can't say that all phase 3 studies are a certain period of time, because it really depends on the study and what they're looking at and the numbers. But depending on the outcome that they're looking at, it can vary within a certain study. Certain efficacy outcomes may be shorter, others may be longer.
Thank you. Another question came in about the hold that went on, and you alluded to it, Dr. Nair, as far as this transverse myelitis and the comment that there may be a multiple sclerosis patient. And as you remember from the swine flu from, I guess that was 1976, Guillain-Barré concerns, these other neurologic concerns. Could you describe a little bit of the process at the FDA for these types of, especially these neurologic symptoms, how a little bit more detail on how that's evaluated, or as far as trying to assess causality?
Yeah, so I'm going to speak in general terms, but these clinical trials are evaluated very, very closely. Even before, the medicine is administered to patients, everything is looked at in terms of what the safety monitoring that's going to be in place. Is it adequate? What are they looking at? And during that period where that close review is being conducted, they'll oftentimes identify adverse events of special interest. And these are in the case of vaccines, many times they're autoimmune conditions, such as transverse myelitis, where we're concerned that there might be some molecular mimicry where, we're triggering an immune response, but there's collateral damage. And so they look at a number of different adverse events that are autoimmune outcomes, and then they ask the sponsor, if anyone has these outcomes, notify us, or there has to be some type of procedure in place to evaluate it. And speaking in general terms, often there's a data safety monitoring board who are separate from the investigators, and they receive this information about safety. Now, the studies are blinded, don't know if the patient received, if the patient has an adverse event, they don't know if the patient received the vaccine or not. But once they receive an adverse event, such as transverse myelitis, that's concerning, they unblind and find out what the patient had, and then decide if any other conditions are met. So, that sort of is a thumbnail sketch of the process. Is that there's close monitoring. If a patient has an adverse event, it's reviewed by a monitoring board. It's unblinded to determine what they received, if they received the vaccine candidate, And then in terms of assessing the causality, it can be very challenging. Transverse myelitis is a theoretical concern. To my knowledge, there's not a vaccine that's been shown to cause transverse myelitis, so it's a theoretical concern. But it's an important one that needs to be evaluated. So they look at the temporal association. When did this patient's symptoms start? Sometimes in radiographic findings, MRI, so forth, you see lesions and you can determine, well, this probably might have been there beforehand, and talking to the patient. You do other studies. Transverse myelitis can be associated with other infections and things like that. So there's a thorough evaluation of causality and what are the chances it's related to the vaccine or if it's related to something else.
Oh, thank you for that detailed response. Another question we have for you, Dr. Nair, is these trials, and from what we heard historically, as far as the number of patients, it seems like there are maybe even more patients than being enrolled in some of these trials compared to the historical past.
Yeah, and I should clarify, I'm not involved in the trial design for any of these products, but it's my understanding that most of them are on the order of 60,000, which is more than some vaccine candidates we've had, and less than others, but is a substantial number, I think. The other thing I mentioned in terms of limiting factors is for studies, and Operation Warp Speed has been able to sort of circumnavigate this one is patient recruitment. Oftentimes recruiting patients for these studies takes a long time. And just the nature of this pandemic we're in, they've been able to sort of ramp up patient recruitment for this.
Oh, thank you. And there was one other question on the statistics from your slide there, as far as how many patients do you need to see a 1% adverse drug rate?
Yeah, I don't want to give an incorrect answer, so I think I'll table that, but I'm happy to go back to our statisticians and make sure I can give a precise answer to that. So if there's I don't know if there's a way to find out who provided that question, but I'm happy to go back and get an answer. But I don't want to give anything that's not 100% accurate.
Okay. Yeah, we can, I think, handle it and post that back up later. So thank you for that as well. Another question came in as far as assessment for durability of the protection from a vaccine. How long is that usually carried out, or how's that determined or assessed in this setting?
Yeah, that's a great question, and I don't know the precise answer. I do know that there's thinking that they do need to test people to see when, how long immunity persists, and it is a concern. But I don't know the exact time frame that they're recommending for these candidate vaccines.
Well, thank you again, and I'm going to turn it over to Dr. Paul Wax for a few. He has some other follow-up questions and a few address maybe a few more of our questions that we have here. We've got lots of them. We'll try to get to as many of them as we can. Take it away, Paul. Yeah,
great. Yeah, thanks, Keith, and thanks again for really superb presentations. Both presentations were really excellent. I have a question again about the timing issue. So it's actually a two-part question. I mean, the first part is, if you enroll 60,000 people into a randomized study, what's the plausible shortest time that you may have, some sort of result? You mentioned that, for certain data points like safety, you may continue to follow these patients over an extended period of time. But what's the shortest time, in a 60,000 person trial, what's the shortest time you might have results? I think you alluded to maybe 6 months, but I just want to hear back from you on that. And then if you could just explain, once there's an understanding that the vaccine may be safe and effective, does this sort of vaccine candidate then go to an EUA process where it's short-tracked to get the approval, and how long does that generally take?
Yeah, so in terms of the efficacy, I think it really would depend on the epidemiology of the condition. If in areas where the COVID-19 is rampant, you might see some difference in efficacy, sooner rather than later. I think the typical the clinical vaccine candidates that are under clinical trial, I don't know what the time frame is that they've established that they're going to unblind and then look at the efficacy. I know that the safety outcomes that they're monitoring at least for 6 months, and for many of them. But I don't know in terms of when they're looking at the efficacy data. There is an FDA guidance that talks about sort of what, and a guidance is sort of what the FDA does if for instance, if hypothetically, if we want to give companies are submitting medicines for lowering cholesterol, the guidance would address, this is what we look at for a medicine that's lowering cholesterol. For COVID-19, there is a guidance that goes through in detail sort of what the FDA's current thinking is in terms of efficacy and safety for a COVID-19 vaccine. That guidance is available on the website and was released in June, and you can be easily found on the FDA website. EUA refers to emergency use authorization. There's a number of mechanisms. The traditional route for drug approval is going through the phase studies and then submitting the data, and then depending on the indication, it's a 10-month review clock for FDA to review after all the data is submitted. With emergency, now there's other ways to accelerate that review clock, but with emergency use authorization, the Secretary of Health and Human Services can issue a declaration that there's a life-threatening emergency. And the threshold for the FDA review shifts. For efficacy, basically the threshold is that it's reasonable to assume that the product works and then the known benefits outweigh the known risks. And so that's a very different threshold than the traditional route of approval, which is substantial evidence for efficacy. To my knowledge, there hasn't been anything saying how long, once a sponsor were to submit, and they have to submit an application for an EUA, how long the FDA would take to review that. I don't believe that that's been determined yet, but the goal would be, if an EUA is submitted, the goal would be to turn that around fairly rapidly while ensuring that, again, it meets that threshold, that it's reasonable to assume that the product is safe and effective. And EUA would be sort of a stopgap. The clinical trials and things would still be going on, and ultimately, we would want these products to go through the full approval. EUA is kind of considered a stopgap in terms of an emergency.
Okay, great. Thank you very much. Another question about the demographics of those enrolled in these trials. Does the FDA require certain demographic representation? Are these trials going to be done in children as well as young children under 2? How do they assure that various racially various groups that are racially diverse are all included in these trials?
So yes, when these protocols are developed, and this is a very important issue, and thank you for asking that question. It's so important that the population that is represented in the clinical trials is diverse, and it's representative of all Americans that are going to be potentially receiving this vaccine, and that's critical. And so it's recognized in the agency that that's important. There's an Office of Minority Health, and then there's a lot of discussion about it being inclusive in these trials and ensuring that there's adequate participation. And so basically it's a lot of discussion with the sponsors of what their methodology is going to be to ensure that they're recruiting patients who are what the demographics are representative of people that will be receiving the vaccines. As far as the age issue, that is a little bit tougher, and sometimes recruiting, particularly with pediatric patients, can take longer, and there's a number of issues. Obviously pediatric patients, with informed consent and so forth. So that website that I cited, the clinicaltrials.gov, it'll show the age range for each of these studies, and it varies. There is an effort to include, because this COVID-19 disease is so strikes the elderly and is so devastating to that population, there is an effort to include people that are older. Sometimes in clinical trials we focus on young, healthy people, but there is an effort to include the elderly in these phase 3 trials, and you can see the age breakdown. Pediatric, oftentimes what happens is the initial, after the studies are conducted in adults, and we have data there, there'll be pediatric studies. That is, in many cases, a requirement for approval that they do conduct some pediatric studies, but sometimes those requirements are put on hold while we get the adult data.
Okay, great. Thank you very much. I'm going to turn this over to Dr. Kazi. I think we'll probably run over 5 or 10 minutes just to try to get to some of the ongoing questions in the queue. Dr. Kazi?
Thank you. Dr. Shepshelovich, I have a couple of questions in the queue for you, so I thought I'd just ask them right now. One of them is related to your study findings. I think you've shown that in one of the slides, but I think some of our participants didn't catch the details of that slide. Could you comment a bit more on that table that you showed and whether you saw some characteristics or variables that increased the risk of some of these safety concerns? And I believe you mentioned that briefly, but can you maybe discuss that again?
No, so we try to look both at vaccine characteristics and perhaps even more interesting for us, for any characteristics of the pre-approval data that might be associated with any post-marketing safety issues. But the only thing which was different, as I think I briefly mentioned, was that vaccines for influenza tended to have less post-marketing safety issues. Other than that, neither vaccine characteristics nor pre-approval nor existing pre-approval data was any different between the two groups. And as I've mentioned, basically, as almost no vaccine had any significant post-marketing safety issues, so obviously it was very hard to find such associations, statistically speaking.
Thank you, and thank you for this answer. I think that takes care of it. The next question is, after completing the study, what recommendation would you make, for example, to the FDA as they plan on engaging this very important process of monitoring the safety of the COVID-19 vaccine?
So first, I'm not sure the FDA needs my advice. I can say that I don't envy people who need to make these kind of decisions. I think on one hand, everybody wants to have a safe and effective vaccines as quickly as possible in the face of this horrible pandemic. I'm actually treating COVID patients daily now. So I more than understand the pressure to have a good vaccine now, tomorrow, now, today. And on the other hand, if any vaccine proves to be unsafe, obviously it's going to be a very bad result, both for trust in authorities, the FDA, this vaccine, and probably in other vaccines as well. So they're kind of between a rock and a hard place. So I don't envy them, and I'm sure basically what the data I've shown shows that these are people who know how to get safe and effective vaccines approved. They have a good track record in that. They've been doing it for tens of years. And I'm sure they want to have a vaccine approved as quickly as possible. And I'm also sure they understand everybody's watching and the importance of having a safe vaccine as quickly as possible. As I've said earlier, I'm treating patients with COVID right now, and if the FDA will have a vaccine approved, I'll be more than happy to take it. I think our data shows that they know what they're doing, and if the FDA vouch for vaccines' effective and safety, I'll be more than happy to take it.
Thank you so much for your presentation and for sticking around for the Q&A. Can I just throw a question for Dr. Nair? Dr. Nair, regarding the rotavirus vaccine that Dr. Shepshelovich mentioned in his study that was identified by VAERS 2 months after the release of that vaccine, had VAERS not been available, for example, or let's say this passive surveillance had not occurred, had this passive detection had not occurred, what would have been other tools that you think would have caught it?
Well, that's a difficult question. It's hard for me to envision. VAERS was established by legislation by Congress and has been up since 1990. So it's difficult for me to envision how we would function without it. There are requirements that the manufacturer, so people, when they receive a product, they have a couple of options. They can submit a form through VAERS, or they can contact the manufacturer, and the manufacturer fills out a VAERS form and submits that data to us. So I guess it's conceivable it could have been picked up by the manufacturer, and that data would have been submitted to the FDA because that's a requirement, and it would have been picked up that way. It's hard for me to say. It's an interesting question. I don't think it would have been quite as the rapidity in terms of picking up the signal would have been quite as effective, but it's theoretically, I guess it could have been picked up at some point.
Thank you. This really highlights the responsibility and important role of healthcare providers in reporting these to the FDA. Thank you for your presentation. I'll turn it back to Dr. Keith Burkhardt.
Dr. Ziad-Khazi. A couple more, I think we have a little time for a few more questions. Again, for Dr. Nair, question about this novel DNA vaccine. And as far as maybe needing more time and the concern about insertion of foreign DNA into a host genome, our questioner is asking, should there be some special concerns for this or extra scrutiny or so?
Yeah, the vaccine candidates that I highlighted currently are RNA, and so although there is concern, it's less concern in terms of integration into the host genome. There is now we do have, I believe down the road there may be DNA candidate vaccines, familiar with those, but we do have gene therapies that have been approved, and we have looked at this issue and issued and so we do have guidances in place looking at long-term follow-up, because that's a very good question, and it's a very important issue. When you have DNA, at least, there is this concern: is it going to be integrated? And even for these studies, as you know, the assays are conducted to look at whether that occurs before it's even used in humans, and then afterwards it is monitored very closely. And those are, at least for some of the gene therapies, where we do long-term studies to evaluate whether there's any issues that arise from possible integration. So it's looked at multiple levels. I'm not as familiar with the vaccine in terms of DNA vaccines. The vaccines I was highlighting were mRNA.
Thank you. A couple other questions in that here regarding post-approval and monitoring again. And you've had your experience in the Indian Health Service. That seems to be a vulnerable population. Do they have their own database, or is there anything special as far as for trying to monitor in that population? And another follow-up, but unrelated to this, but social media monitoring, is that something that CBER's involved with in that as well for monitoring for emerging adverse events?
Yeah, I'm not aware of any anything looking at social media. The CDC may have something in place where they look at social media. I've read sort of articles that might be a helpful tool in terms of. I've read it more in the context of outbreaks, actually, that tracking the outbreak using Twitter or other social media platforms. But to my knowledge, I'm not aware of anything looking at social media in terms of tracking adverse events. Indian Health Service, again, a really important question for that population that has been very hard hit by this pandemic. And I don't want to speak for them. I don't know if they have their own kind of data collection looking at that. But it's my understanding that there have been discussions with CDC to sort of ensure that population, that we are tracking the safety in that population. I don't know the details of that. And one thing we do is data sharing. I should point out with VAERS, that data is publicly accessible. It's been de-identified, to prevent privacy concerns, but there's a program called Wonder. Wonder is the public-facing aspect of VAERS, and so you can see, you can track, for certain vaccines and things, what reports have come in, and so it's fairly, I think, straightforward to use Wonder, and it is public-facing.
Thank you. And follow up on the VAERS, has there what has been the history and trending in that as far as reporting and that into the system? I know with VAERS there's not as much public reporting into that. Is it the same with VAERS, or does that get a little bit more from the public, more so than healthcare providers or the manufacturers?
Yeah, it's somewhat inverse. VAERS, as I understand it, most of our reports are from the public, and fewer from the manufacturers receive fewer reports. So, and as with all these systems, which is good, as they get more accessible, we're getting more increase in submissions. And we anticipate with a COVID-19 vaccine, we're anticipating an increase in the number of reports that we'll be receiving. So we're sort of ramping up in anticipation of that.
And okay, I'm going to turn it back over to Dr. Paul Wax for, I think, a final couple of questions as we're approaching quarter after the hour here. So, Dr. Wax, please.
Yeah, great. Yeah, thank you very much. So, just two last questions. The first one, I'm not sure if you have the info, but there have been some reports that have come out that in some of the developing countries in Asia and elsewhere that they've been using increased amounts of the oral polio vaccine to perhaps give the immune system a boost that may provide some benefit in terms of COVID-19. Do you have any info on that, or other vaccines in general kind of helping the immune system as we wait for the specific vaccine for COVID-19?
Yeah, I've seen that data, but I'm not familiar with anything that's been submitted to the FDA looking at that specifically.
Okay, great, thank you. And my last question, which you probably might anticipate nowadays, is could you just maybe provide us with some comment or even reassurance, perhaps, about any sort of firewall that's in place between the science and the politics? What's been described in the past, vaccine development and whatnot, seems to be all scientifically driven. At least that's the impression we're left with not much political interface. But now all you need to do is turn on the news 24/7, and this is on the tip of everyone's tongue. So is there anything I realize it may be a tough question, but is there any way you can just address the scientific integrity of your mission and, the attempt to keep the politics away?
Yeah, thanks for the question. I think it's, I would stress, as I said at the beginning of this talk, my talk, these are my opinions only. I'm not expressing anything on behalf of the department or the federal government or FDA. My feeling is that the process is scientifically based and that it will continue to be based in science. And I think I've tried to, and I'm kind of kicking myself. I didn't highlight this in the slides, but I tried to verbally highlight that this process is very transparent. We have a guidance that's posted that says this is the criteria we're looking at for a vaccine in terms of safety and efficacy. And we're going to have public meetings. The CDC's had several public meetings where we have these outside, really world-renowned subject matter experts that are advising CDC and FDA on what to do. And then our reviews that our division conducts and that other divisions, those are released publicly. That sometimes some information is redacted for privacy and commercial confidential information. But for the most part, our reviews that our medical officers do are released and posted on websites for people to digest and look at. So I think in terms of politics and science. For me, I think the process, maintains its integrity, and I think transparency is always the safeguard to ensure that. And I think it's a very transparent process, and it'll continue to be a very transparent process. It'll be easy for people to say, well, you deviated from your guidance. It'll be easy for people to say you deviated from the advice you received. And we'll have to answer to that, and as we should, because we work for all of you. We work for the public. And so that's my view. There was a editorial, I mentioned that the FDA is broken down by centers. There was an editorial written by our center director, Dr. Marks, and several other center directors, and collectively they had over 100 years of experience at FDA. And it appeared in USA Today, and it touched upon this topic and highlighted the importance. These are career government officials who have been in public health their entire careers for decades, and it highlighted their approach to COVID-19 vaccines and treatments and the integrity. And so that's available on USA Today. I'm not putting a plug for USA Today, but that editorial, I think, is worth a read for people that are interested in that issue.
Well, thank you very much. Again, I'd like to thank Dr. Nair and Dr. Shepshelovich for two really outstanding presentations on an extremely timely topic. And appreciate all the wonderful questions that we got from the registrants today. Next slide, please. This webinar has been recorded and will be up on the ACMT website within approximately 48 hours, and we'll send out a notice to registrants about the recording so they can access it. Next slide, please. We will be continuing this webinar series, which since March has been practically every Wednesday. In October, we'll go to every other Wednesday. But the next 2 Wednesdays, we'll be continuing on the weekly basis. And next week, on September 23rd at 3 p.m. Eastern, we'll be having a webinar on contact tracing. Dr. Crystal Watson from Johns Hopkins Health Security Center will be talking about the latest on this very important public health process of contact tracing. And then we'll have a shorter talk from Dr. Stephanie Hahn from the Georgia Poison Control Center, who will be talking about some practical implications of contact tracing. So that's next Wednesday, September 23rd at 3 p.m. Eastern Time. Next slide, please. This prospectus is available on our website. It does list all of our prior webinars. We have quite the collection now, as well as upcoming webinars. We encourage you to spread the word about the webinar series. It's wonderful that we can get such a large audience. We had close to 900 people on the webinar today, so much appreciated for you to take the time and learn together about the latest on COVID-19 so we can be better prepared. Thanks again, and have a good day.